Hello everyone. Today I am going to demonstrate gluconeogenesis, and this is the part 1 of the sequel. Key points that today we are going to learn. Number 1, what is gluconeogenesis? That means the definition of that. Then we will know what are the sites of gluconeogenesis. Then what is the reaction sequence of the same. Then how amount of energy is expended during this gluconeogenesis. Then significance of the gluconeogenesis, and lastly the regulation of this gluconeogenesis reaction. If you are new to my channel, don't forget to subscribe, like and share my videos. So what is gluconeogenesis? Gluconeogenesis is a process of formation of glucose from non-carbohydrate substances. That means when the glucose is prepared from something which is not a carbohydrate, that is called gluconeogenesis. In human being, lactate and pyruvate are two quantitatively the largest sources of glucose through gluconeogenesis. These two helps mostly during exercise. Means when a person undergo exercise, then the muscle produces the lactate. Pyruvate is also produced in our body. Now this lactate and pyruvate is converted into glucose for the reutilization by the muscles by the process of gluconeogenesis. During starvation, glycine and alanine, the glucogenic amino acids, are considered to be the major source of glucose through this process. Smaller amount of glucose is also found to be synthesized from glycerol. So, in the human being, there are 5 major sources of gluconeogenesis, and these are: glucose is formed by gluconeogenesis from lactate, pyruvate, glycine, alanine and glycerol. In case of ruminants like cow, propionate from cellulose fermentation forms a major source of glucose, but it is in the case of ruminants only. Now we will know what are the sites of gluconeogenesis. There are 2 main sites of gluconeogenesis where the gluconeogenesis actually happens. One is in the liver and number 2 is in the kidney, mainly the renal cortex region of the kidney. If we compare between the capability of liver and kidney regarding the gluconeogenesis, we will find that the liver normally synthesizes 9 to 10 times more glucose by the process of gluconeogenesis than kidneys. So the liver form more glucose by the process of gluconeogenesis. Now we will learn the reaction sequence of gluconeogenesis. So, first of all, we have to know that gluconeogenesis occurs in 2 different parts of the cytosol cell in combination. One is cytosol part and another one is mitochondria part. So, this process starts with the lactate, which is accumulated in the muscle. Lactate is converted into pyruvate with the help of lactate dehydrogenase enzyme, and in this case you can see that some transfer of energy happens. Now pyruvate enters from cytosol into the mitochondria. This pyruvate with the help of ATP and carbon dioxide is converted into oxaloacetate and inorganic phosphate, as well as ADP comes out. Now this reaction happens with the help of pyruvate carboxylase, you can see. Now this oxalate with the help of the enzyme PEPCK, it converts into phosphoenol pyruvate at the cost of GTP, and carbon dioxide comes out. Now this oxaloacetate on the other hand is converted into malate with the help of malate dehydrogenase. This phosphoenol pyruvate from mitochondria enters into the cytosol. There, it by a both way reaction, it is converted into 2-phosphoglycerate with the help of enolase, and water comes out. Then 2-phosphoglycerate converts into 3-phosphoglycerate with the help of phosphoglycerate mutase. It is again converted into 1,3-diphosphoglycerate with the help of phosphoglycerate kinase at the cost of ATP, of course. Then this compound is again converted into glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate then directly converts into fructose 1,6-bisphosphate with the help of aldolase B, and dihydroxyacetone phosphate comes out. This fructose 1,6-bisphosphate is converted into fructose 6-phosphate. Water helps in this reaction and inorganic phosphate comes out, and this sequence occurs with the help of fructose 1,6-bisphosphate. Now this fructose by some reactions ultimately converted into glucose. Now we have to learn something about malate. Like phosphoenol pyruvate, this malate also comes out from mitochondria into cytosol. Here by some reaction it converts into oxaloacetate, and this oxaloacetate and phosphoenol pyruvate, they are interconvertible. This malate converts into oxaloacetate with the help of malate dehydrogenase, and oxaloacetate with the help of PEPCK converted into phosphoenol pyruvate. Now this fructose 6-phosphate is converted into glucose 6-phosphate with the help of phosphohexose isomerase. This glucose 6-phosphate ultimately converted into glucose, which is the main or the ultimate product of gluconeogenesis. The enzyme which helps in this conversion is glucose 6-phosphate. Water enters, inorganic phosphate comes out. So this is the sequence of gluconeogenesis. Now, energy expenditure during the gluconeogenesis. 6 high energy phosphate bonds are spent in the gluconeogenesis of one glucose molecule from 2 lactate or pyruvate molecules. Means for 2 lactate molecules, one glucose molecule is found, and 6 high energy phosphate bonds are spent. How? So, energy spent for one lactate molecule, we can see in the table, the reaction steps and the phosphate bonds spent are given here. This pyruvate when converts into oxaloacetate at the cost of 1 ATP. This oxaloacetate again converted into phosphoenol pyruvate at the cost of 1 GTP. 3-phosphoglycerate is converted into 1,3-diphosphoglycerate at the cost of 1 ATP. So 1 ATP plus 1 GTP, ultimately it is 3 total. So energy spent for one lactate molecule is 3 ATP. Now energy spent for 2 lactate molecules, 3 into 2, 6. So total 6 high energy phosphate bonds are spent in the gluconeogenesis of one glucose molecule from 2 lactate or pyruvate molecules. Now we will see the significance of gluconeogenesis. Gluconeogenesis maintains the liver glycogen and a steady basal level of the blood sugar during starvation. Starvation means it's a condition of the body when for a certain amount of time the people doesn't take any food, but still there are some parts of our body which are very much required of sugar even during starvation. So during the starvation it maintains the supply of glucose to the brain, the kidney, the red blood cells and the body muscles. So these 4 are in high requirement of blood glucose even during starvation. The next important significance of gluconeogenesis is it maintains the Cori cycle. Now, what is Cori cycle? Lactate produced by glycolysis in the released into the blood. This carried to the liver and reconverted by gluconeogenesis into glucose to deliver the muscle so that muscle can work more vigorously. In third significance, we will learn about a disease of the infant, which is methylmalonic aciduria. Now, what is this? Gluconeogenesis serves as the major metabolic pathway for propionate. Genetic failure of gluconeogenesis means if gluconeogenesis cannot happen in the body due to some genetic defect from propionate in infants causes a fatal disease which is known as methylmalonic aciduria. So, these three are the main significances of gluconeogenesis. Regulation of gluconeogenesis. Gluconeogenesis is regulated mainly by some enzymes and by some hormones. First of all, we will know about the regulatory enzymes of this pathway. Pyruvate carboxylase, phosphoenol pyruvate carboxylase, then fructose 1,6-bisphosphatase, then glucose 6-phosphatase. So, these four are key regulatory enzymes of this pathway. Now, the hormones which control this gluconeogenesis are number 1, glucagon, which is released from the pancreas. It increases the gluconeogenesis. Adrenaline also increases the rate. Glucocorticoids also helps, but insulin, it decreases the rate of gluconeogenesis. So, these two hormones, this glucagon and insulin, both comes out from the pancreas but have different effects on gluconeogenesis. Glucagon increases the rate of gluconeogenesis, means it helps to happen the gluconeogenesis, whereas the insulin, it decreases the rate, means it obstructs gluconeogenesis from happening. Some references are there if you want to know more about gluconeogenesis. So, there are 3 references you can go through to learn more. Thank you very much for your patient listening. Part 2 is coming, where we will discuss about the question answers on gluconeogenesis. So long. Bye bye.